crisis. It's related to wine and cheese in France. If you have a problem, it should be a national crisis, of course. But if you see the numbers, over 100,000 acres, which is 12%, of the production in France is right now compromised, and it's not economically profitable. It doesn't mean that these blocks are all dying and they are not economically profitable. There is a time that the grower needs to pull out because they're not making any money. So to give you an idea, these diseases, and that's why it's so complicated to work with these diseases. This is the vineyard age, and this is the percentage of incidence of the disease in the vineyard. So we see that the older the vineyard gets, the more disease we are finding in our vineyard. This is not something that you are going to identify immediately since year one. Some of these fungi are actually acting inside the wood, and we may not see any symptoms in the plant until 5-6 years later, when the problem is already inside the plant. To give you an idea, in blue, that would be a healthy yield. So after the third, fourth year, you can reach your 10 tons per acre. These are data from California. But you can see here, after the 5th year when we start seeing disease, how the production is declining. And this is, as I mentioned before, totally economically unviable. So in mid-2000s, I would say, BC started to experience, important yield and plant losses. At the beginning, of course, primarily attributed as a biotic factor, winter kill is a problem for us, spring frost, or some diseases that we know happen because of, our conditions where we grow grapes. We have crown gall, which is a bacterial disease that is actually favored because of, cold conditions. But the industry, was significantly concerned about some of these problems, so they wanted to really, what's going on? We didn't know anything about this problem in British Columbia, so they really wanted to understand the status of this disease. So here we come. The plant pathology team in the Summerland Research Station. We started a project actually in 2010, and these were actually our main objectives of the project. First, as you go to the doctor and they need to figure out what you have to treat you, we need to know that. We need to know which pathogens are associated with the problem in British Columbia, the incidence, the spread of those pathogens. We work a lot in implementation of molecular diagnostic tools, DNA-based tools, to detect these pathogens, and to work in our research, and also to transfer. We have a mandate of developing this type of technology to transfer to other laboratories or another research agencies. Understand pathogen and disease epidemiology, which is for me one of the critical and most important parts, and we'll get to that, with of course, the main goal of implementing control studies, which, until recently, there was absolutely none here in British Columbia. So we started with a very massive field survey in all the regions in BC, and Kamloops will be also here because we have been here in Kamloops collecting samples. So over 200 vineyards visited, more than 60,000 vines inspected. We were collecting samples from all those vineyards to bring to the lab. One of the, I would say, shocking things we saw is 90% of those vineyards we visited, they had disease in the vineyard already, whether they were young vineyards or mature vineyards. So the disease was very well established already. And this doesn't mean that all the vineyards have the same incidence. We have vineyards with 2-3% of the vines infected. We had vineyards with 80-70% of vines infected. So there was a huge variability in infection, but we can see the impact. Overall, in all our research products, we saw about 10% of the vines infected and starting declining or collapsing because of these diseases. And 10%, you may think, well, is maybe not too much. It's significant. For example, a grower, when he plants a vineyard, he can expect maybe a 1-2% of losses of vines that they may naturally die. But something about that, you start getting really a hit, on replacing those vines or not getting, crop from those vines. And these are only the vines we could see symptoms, actually. And as we know, with the disease I explained before, there were probably many more vines that the infection was inside, but we were not able to see, the symptoms in the outside. So this is a typical situation you can see, and you can observe, all the losses that this grower is experiencing because of the disease. So the idea is to tell you is not only the money that the grower has to spend in replacing those vines are going to take 3 or 4 years to get to production, but also this is a nightmare for the grower to manage. It's a completely uneven vineyard. How are you going to irrigate now those vines? Because those vines are going to need more, more water than these ones, but you have one line, to irrigate the entire row. How are you going to spray those vines? So there are more issues than just, replacing those vines. So overall here, if we collect all this is the figure showing you all the different blocks we surveyed with the disease incidence and the vineyard age. So it's every single point, and here you can see the average of all those age parameters. We can see between 0 and 5, significant amount of vineyards with significant amount of disease. That's what we call the young vine decline. All those vines that are suffering the problem at the beginning. We have kind of a plateau, until between the 5 or 10. That's most of the vineyards that have been infected and the diseases start progressing inside, but we still don't see really any symptoms or effects. But you can see that after 15 years old, most of the vineyards, they have a significant incidence. And the older the vineyard, the more disease, And that's the problem, There are some diseases we can identify directly and see the symptoms in the field. We can treat them, we can control them. When we see the impact of the disease, it's too late because the infection is already in the plant. So for our first objective, of course, we use traditional, methods for identification. We try to re-isolate directly from the symptoms. We use different culture mediums, and, taxonomically, morphologically, we try to identify the pathogens at least to a genus level. But of course, we have been using a lot of molecular tools. And one of the main problems we have is we cannot just use one single gene, or locus to identify these pathogens. They are so close related, some of them, that for many, we need to do many different loci, to go to species level. Have a multi-gene approach, to find those differences to get to species level. So this is an example of things that we were finding in Okanagan. This is for the disease Blackfoot. This is just the taxonomically work we do, where we, look at the colony morphology, the spores. We can identify some of the pathogens based on morphology, but we do a lot of phylogeny. This is a phylogeny based on 3 different loci, 3 different genes, with the 5 different species we found in the Okanagan. Why this phylogeny is important? Many times we don't really know the species, or they are new species. And in this case, we were able to describe 2 new pathogens here in BC, that they were new to the scientific community, and they were causing problems here in the Okanagan. And those are the descriptions we have. So we use this to really nail down the species and the identification of what we have. So far, we have found more than 40 different pathogens associated with this disease in British Columbia, which from a control perspective is really a nightmare, because you are not going to find one single product that are going to completely control these taxonomically diverse pathogens. They are in different families. This is the family Botrytis, Dasypaceae, Phomopsis. So that's a challenge. This is not, for example, powdery mildew of grapes. It's one disease, one pathogen. We have a lot of chemicals, a lot of biological control we can use. There is a big challenge for that. And with challenge, we try to work also how difficult it is to identify these pathogens. If you are dealing with 40 in BC, overall we have over 100 different fungi causing this problem. You really need to have very good taxonomic skills and know your mycology just by looking at the plates and those spores to know what they are. And it's very, very challenging. I've been working with this pathosystem a lot, and I still having trouble, to differentiate many of them. So one of the things we have is the lack of accurate methods, diagnostic tools, is what is really, slowing us down in developing these control strategies. So one specific project we have, it was to try to develop and implement a multiplex system that will allow us to identify as many of these pathogens as possible in just one test, which is really, that was really the challenge, to have this multiplexing approach. So for this reason, that's when I joined actually also part of my postdoc research. We decided to go with a DNA array. For those who are not very familiar, this is not new science or rocket science. It has been done way, way back in the 90s, mostly when they started to work on identifying mutations for cystic fibrosis. But it has been lately applied and implemented in agriculture. And we thought that for our purpose, that was the best match we could, obtained. So what is the DNA macroarray? So basically we have a nylon membrane where we have here very specific probes, and when I say specific, these are species specific. So this probe is going to detect just one species and not the others. Those probes are linked to the membrane with this amino group, and what we did is we tried to get as many diversity as possible from pathogens all around the world. We didn't want to identify just the pathogens that are causing the disease in BC, because we know there are diversity within the species as well. So what happens if these pathogens come in planting material? We want to identify a species that is not only in BC, but it's also coming maybe from Europe or from the States or from somewhere else. So we were able to get over 140 isolates and 72 different species in total. So what we do is we target a specific gene. In this case was the beta-tubulin gene, because for all our different species, that was the most informative we could go. And basically we sequence all those isolates. We do a lot of analysis and alignment. And we were looking these different polymorphisms, to develop our specific probes. So it is one species in the next entry. You can see here all these same bases, but you can see how they change for the different species. So we were looking these differences to develop our probes that are going to differentiate exactly one species from the other. All those probes, they get into the membrane, and we get a sample. What we do with this sample is we start total DNA. We label the sample with these big 11 UTPs, and if the sample contains our species that we are looking for, there is a reaction, a match, and by chemiluminescence or X-ray, we are able to detect it. So basically what you are seeing here is all our controls, and these are the probes for one specific species. And our goal is to see there is no cross reaction, so every dot here is a single identification of one species. So for example, we have here one of the pathogens in the group Phaemonella. Phaemonella chlamydospora is one of the pathogens we have for plant diseases. This is an isolate from South Africa, and also this is an isolate from British Columbia. This is an isolate from California, and an isolate from Italy. So we develop a system that we are actually able to detect the same species, from different regions. And these are other species within the same genus that we are able to identify and to see there is no cross-reaction here. So with this system allow us, to detect up to 70 different species in just one test, which is quite significant. We try also to look into how low in detection we can go. So based on these two pathogens, we did some serial dilutions, and we can see that it's very comparable to PCR, but actually, we could actually detect one step further, in the plant, in the soil, or in water. We can use this system to identify pathogens for many different things. So we implement this system. These are, for example, planting materials. We know these pathogens can come in the planting material from nurseries. So one of our targets was to use the system we developed to see the healthy status of planting material coming into Canada. Because we import a lot of vines, mostly from Europe and North America. So we did a very large study with different nurseries, and we found basically, as you can see here, that up to 92% of the plants coming into Canada would host, some of these species. These are all the different battery of species that we were able to identify with planting materials coming here. So this is not to blame the nurseries, and to say all the fault is in the nurseries, but this gives you an idea of what we need also to focus on, to start working on, to fix the problem, to see how we can help nurseries, to control the issue at the nursery level, so we are getting cleaner and cleaner plants. So another very interesting thing we got from this research is we were observing that we were able to identify pathogens in asymptomatic plants completely healthy, from nurseries. And you can see here the symptoms. These are necrotic symptoms, and we were basically isolating the same species. So this tells us that many of these fungi can be already in the plant, not causing any problem, as we have been talking before, but there is some change in the plant that they are becoming these fungi to be latent pathogens or endophytes to true pathogens. And this is a line of research that we are actually focusing now. So this was very, very interesting because, for example, in 2017, it was a big change in our industry in Canada, in terms that the Canadian Grapevine Certification Network was formed. It's formed by all 4 growers associations from the main grape growing regions in Canada: BC, Ontario, Nova Scotia, and Quebec. And the main goal of the association is try to provide growers with the cleanest plants possible, free of these pathogens and other ones, for example, like viruses. And we have been playing, and we are still playing a big role on that. I'm part of the scientific advisory committee for the CGCN, and we are trying to work with them. What we need to do, what we are identifying to try to reach this goal. So one very important thing, and I go through the third objective, is so we know which pathogens are dealing with in British Columbia. We know the incidence. We know it can come with the planting material. We have developed good diagnostic tools. So now my goal is, well, we are not in California, we are not in Chile, we are not in Europe, our conditions are different. How this disease or these pathogens work under our environment to identify the inoculum availability in the field. We know that these diseases go into the pruning grounds. So my goal is, when these spores of these fungi are in the environment, so I can tell the growers to target the control strategy. So we want to identify periods of low risk of infection, where maybe cultural practices or maybe working with the pruning can work to minimize the infection. Periods of high risk of infection, where we may need to target our other type of control, chemical control, biological control, and the environmental factors that favor this spore release. The idea would be to have a model that we can build, and we can actually tell the growers which conditions are optimum for these pathogens to be in the environment, so they can start triggering the control strategies. This is the life cycle of overall life cycle of these pathogens. So we have here our symptomatic vine infected. If we go inside, we are going to see our fruiting bodies. So these fruiting bodies of the fungi are embedded in the bark. So within these fruiting bodies, we're going to have the spores. Those spores are released depending on environmental conditions. They are going to colonize the pruning grounds. Our infection is going to start and continue, and then closing the cycle. So it's very important to understand which environmental conditions are favoring this spore release, and then the host response, when the pruning grounds are going to be most susceptible, so these spores can land and infect. So I have a couple of videos that this shows this, I think, very good. So what you're going to see now is one of these fruiting structures. As you know, in fungi we have asexual and sexual reproduction. So asexual is more a clonal, the spores, the conidia, and we will see later the sexual reproduction, which is a little bit different. So I'm going to show you here where all this, this is a vineyard. Our fruiting bodies are going to be embedded in the bark in areas that are already infected, So we go to our cordons. So these areas that can be infected, all pruning grounds. You can actually see with the naked eye, with the lens, you can see these little black dots. What's happening? Those fruiting bodies are waiting for the optimum moment to release the spores. They don't want to release the spores for nothing, to don't have the effect of continuing the life cycle. So an important environmental factor is humidity and rainfall. So we have actually here seen the fruiting body. I don't know if you can see a little hole they have at the end. That's called the ostiole, and that's where the spores are released. So when the fruiting body, feels that the environmental conditions are optimal, we are ready. It's already discharging the spores here in the cloud. So you can see here the thousands of spores that go out of that one fruiting body. These spores, they get disseminated in the environment, and they're going to start infecting other fruits. So this is the asexual part. Where are we going to find this fruiting body? In all cracks, all pruning grounds. That's where our fruiting bodies are going to be. This electron microscope of one of these fruiting bodies embedded. We have this long neck with a hole at the end. The morphology of these fruiting bodies is very diverse. It has so many different fungi, but this is just one example of other different type of release which is a sexual release. So at the end of this neck I saw you with this hole, you're gonna see the spores coming out within a sac. This sac we call ascus. It's a very, very thin sac with 8 spores inside. And this is very typical of the group of ascomycetes that are the main pathogens, having 8 spores within that sac. So again, when the environmental conditions are proper, the fruiting body is smart enough to say, okay, I'm gonna discharge my spores because they're gonna have the success of germinating and infected. When those sacs are ejected, because of a difference of pressure between the inside of the sac and the outside, these sacs breaks and it gets all the spores released. Those spores can be taken by droplets of water, by wind. Even it has been a very, very interesting study down in South Africa. They have, I think it's up to 40 or 50 different types of insects can carry those spores in the legs. Okay? So those spores can be spread very, very long distances by the wind. And again, we'll find a pruning wound and continue the infection. So it has been a lot of work done on epidemiology and try to understand, in different regions, because as you can imagine, it's not the same, our conditions are in other grape growing regions. But most of these studies, they have been done using, different systems, trying to read the spores available, in those glass slides put on the field. They are very time consuming, and you really need to know your taxonomy of the fungus to identify which spores are there. So we wanted to go a little bit a step further and try to develop a system that we don't need all that, taxonomy or skills. So try to avoid this time consuming part of the research and develop a molecular tool that we can really collect those spores, extract DNA, and identify. Not only identify, but also quantify. So many of you probably are very familiar with this. It has been this quite recent technology. It's called Droplet Digital PCR. It's kind of the next step of the qPCR, quantification PCR. And the main difference, I have here is with the Droplet Digital PCR, what we do is we break our sample in, 15 to 20,000 different droplets. That's why it's called Droplet Digital PCR. So instead of having one reaction with your qPCR, and increasing that reaction, you are actually having 20,000 reactions in one tube. And in each single reaction is happening, a PCR reaction. And you can have a total quantification, an absolute quantification. So what will we do? We have in the Okanagan Valley 5 different spore trap devices. These are the spore trap devices that they collect air 24 hours, 7 days a week. We have them now for 3 years. We change them weekly. All the air goes into these Eppendorf tubes. And we have in all those areas also weather stations and monitoring. We have our challenges, and you can imagine the Okanagan when you have to go every week to collect those tubes, but we were very well equipped, so that was our exercise, weekly exercise. Another challenge is you really need high-skilled workers to collect them. Really, really, people who knows what they're doing. Anyway, so basically every week we're collecting our tubes from the 5 different locations, extract total DNA. We didn't even look the spores that they were in those tubes. We have a system developed that we can extract the total DNA, run it through the system, and quantify the pathogens we were interested for. So we developed specific probes for specific pathogens that we were quantifying using the system. So what I'm showing you here is one of the sites we have in Osoyoos in southern Okanagan. Here you have the total number of spores per week. Each of these bars are the total number of spores per week. This across 3 years. Here we have the temperature, and you can see really clear, our summer, fall, winter, spring, summer, and the precipitation. So the idea is to understand first when the spores are in the environment, which conditions are favoring the spore release. So we observed that overall, our first discharge of the spores happened end of winter, early spring. And we can see here, end of March, April, that's when we have the first significant detection of the spores. It was very interesting that about a month later, we have the major discharge of the spores. Once the fruiting body is empty with these spores, it needs to regenerate. It's not going to be continuously, So it needs a time to regenerate the spores and continue. So basically, the home-tail message here is almost through the whole growing season, which is not good news for us. There were spores in the environment, And we can see here that during the winter, basically our spore catching is none. It's very, very, very low. One thing we observed is the spore release, it was not so associated with rainfall that as we thought, but it was more correlated to temperatures. And as soon as we were reaching above 5, 6, 7 degrees Celsius temperature, that was kind of the threshold that we saw that spores can be released. So we identified, they were detected mostly from March, April to September, October. First significant spore release, end of winter and spring. That's what I mentioned before. Primarily when temperature is above freezing. Correlated, that not always, to rainfall. And that's why something we are also working right now and see other aspects like relative humidity, dew point, wind, what is also affecting this chart. So, we were trying to get all the pieces of the puzzle together to develop, our best strategies for pruning boom protection. We need to protect those booms, so once the spores are outside, they cannot infect the vine. We have right now not a product that we can spray in the vine systemic that can actually stop the canker, stop the infection inside the vine. So that's when I come to my last point, the effective control strategies for Canadian growers, both chemical, biological, and cultural practices. First, talking about chemical control, we don't have any product registered in Canada, and that's one of the main issues for growers. They have nothing to do. There's not a single product registered for plant diseases right now. So there is a big problem they are facing, but nothing really they can work on. So our first approach was to, screen for many different products. We were looking for products that they were already registered in Canada. They were registered also on grapes. So, if they work also for these diseases, they can do a label extension. So, they can put another disease on the product, and they don't have to go for entire registration of a new product, which is very, very costly. However, we also include products that we know they are working in some other countries and they're registered in other countries to see if they work also under our conditions and the companies are interested in registration here in Canada. So this is the battery of products we have. So we established first, a system in our greenhouse before moving this to the field, because it's a lot of work, to get this product screen. You want to be sure that you are working with the best products before moving to the field. So we have 12 different products. We screen 10 different pathogens. So we have over 3,000 canes rate. And what we have here is in our greenhouse, we have these beds. We put a plastic, we flood the beds. So the water is contained with this plastic. We put this styrofoam. We made these holes, put our dormant capping. So basically, we are trying to recreate a vineyard in our own greenhouse. We do an artificial pruning cut, so we are simulating our pruning gun. We apply our product, our protection for the wounds, and the day after we come with our inoculant and artificial inoculate to see the effect of protection of those products. We come back to this a few days later, we try to re-isolate, and in those that we can re-isolate the pathogen, obviously the product didn't have any effect on the pathogen, but in those that we see nothing growing, basically the product was avoiding the pathogen to come. So among all the different species and all the different products we had, we identified 3 or 4 products that they were very promising, they were working very good. And the best news is we had some products that they were actually working for almost all the different species, which was really, really good. It's very, very difficult to find the silver bullet for the amount of pathogens we have. So we have 2 or 3 products right now that we know they're going to be very good for most of the pathogens. So our next step is moving to the vineyard and the natural conditions, with the product that we know they are working. We are trying to provide as many data as possible so the companies can use that data for registration, and the growers, hopefully in a short time, they will have products available. But I see also, you can see that the pathogen of the inoculum is in the environment for a long period of time. It's not going to be just one time infecting the pruning gun. So we need to be sure that whatever we are putting in our pruning gun is going to last at least certain time of protection. It's not going to be like 24 or 48 hours of protection. So that was another challenge. You don't not only need the product that works for many pathogens, but you need the product that stays a long time on the pruning gun. Or you may need to apply several times the product. So for that, we did some experiments where we what we did with this specific product against one of the most virulent pathogens. So this is our check, our control with no treatment on the pruning gun. So we got almost 100% infection. And what we have is we cut those pruning guns we apply our product, and we go certain number of canes, we inoculate 24 hours later. So this is our 24 hours. Then we come to another set of canes, we inoculate it 7 days later, and then 21 days later. So the idea is to see if that product is still effective in the pruning gun if the spore comes 21 days later. And we can see here that, of course, the day after we apply the product, highly efficient, 100% control, but the product is losing efficiency with time. But still, we can see that even after 3 weeks, we are getting, over close to 80%. So we are pretty sure that with this product, the grower could go maybe in a 3-4 weeks interval of spray, which maybe with 2 sprays in the season, it will be enough to protect the plants. We are also working a lot with biological control agents. These are Trichoderma genus, are known for being biological control. And you can see here a different type of graph. You can see here 24 hours after 100% control, the chemical product decrease with time. With the biological control agent, you have less control 24 hours later, but increase with time. Why? This fungi, Trichoderma, we apply in the pruning wound. It needs time to colonize the pruning wound. It needs time, to completely cover the pruning wound. So when we go and apply our inoculant, our pathogen, that wound is protected. So that's why after 24 hours, our results are not very good, but we can see that 21 days later, we reach the same type of control that with the chemical, with the biological agent. So the question here is, well, when is the best time to apply our biological agent? So we are sure that, we are going to minimize the risk of infection during this time, where the biological agent still is not ready, to give us all that. So I have right now a master student working with me, and what we are trying to do also is to identify native biological control agents in the group of Trichoderma and Bacillus. We have done big search on different hosts. So we have actually identified 7 different species, including 1 new species that we are actually screening for biological control, because that's the thing. If we bring biological control, let's say for, I don't know, very warm region in Spain, growing grapes, and we try to, it may not work, because that particular isolate is adapted to those conditions. And then we are trying to grow that isolate here in March, where we still be at 5-6 degrees Celsius. So it's very important to try to develop this work with local isolates. So we have done a lot of preliminary work, and we have a screen over, 30 different isolates in the Trichoderma and Bacillus. We have this double dual assay. It's very simple. We have our control. We have our Trichoderma, the pathogen. We measure and to see if we have inhibition of the mycelium. So that's the first indication that it can be a potential biological control. So from this screening, everything we have more than 50% mycelium inhibition in this assay, we can move it further, into the greenhouse, doing the same type of experiment. And those isolates that are working, we are moving them to the field. So right now we have 2 isolates that they are really, really very, very promising and working really good. And we are going to start trials now in April in the field. Another thing we have done is try to understand how we can manage the pruning. If we go back to that graph, you have seen that in the winter, there is almost no spores in the environment. And most of our first spores come in March, in late spring, late winter, early spring. Most of the growers tend to prune later, in Turk and Ireland. Our idea is, what if we prune earlier? What will happen if instead of pruning later, which our wounds are going to be made closer when the spores are, what if we prune in December and November? Those wounds are going to have much time to heal under natural conditions. So when the pathogen comes in March, we are going to have less infection. So that's basically the experiment we did here. We have 4 different pruning times: December, January, February, and March. And we came here and we inoculate on March 16, all these different cuts. And this here, the percentage of pruning with infection. So you can see here that the difference between pruning in December versus pruning in March, we reduce infection 40% just naturally by pruning. So what happened? In March, if that wound is open, freshly open, the pathogen comes the day after, the wound done in December, it has 2 months, to start slowly healing. The process of healing of the pruning wound is directly related to heat, and that's why during the winter it's very slow, the healing process. But basically it's a natural death of the surface of the wound, and the pathogen cannot. They need leaf tissue, to start infection. If we go, for example, what happened in April, the second year, if we prune in January, in December we are able to reduce close to 50%. This year in January, 80% less infection, versus pruning in April, almost 100% of the wounds were infected because the wounds are very, very susceptible. So we know that we can manage the pruning in Okanagan and naturally reduce the infection. We are not going to be able to 100% control, but we are trying to give as many options to growers, and to minimize the impact of the disease. So basically, wrapping up, our goal, in the federal government, the research response to an important threat to the industry. We have an industry priority, big problem, big issue, economic impact, no control strategies. So we went through basically all the most important pillars to be able to start giving grower solutions. Identification, characterization of the pathogens, development, implementation of molecular detection tools that are helping us to really speed all the process we are working on, and the epidemiological studies, which I think is key to really understand how the pathogen works under our conditions. Many times you see that, well, let's see what they do in California, and I do what they do in California. Well, we are not in California. We are in BC, and our conditions are totally different. So what they do there, it may work there, it may not work here. Some of the things they do there may work here. But that's why we really need to work in a region-based, strategy. So basically, this research project is giving the first, strategies of control for these pathogens to growers in Canada. We just got funded for a big grant within the CAP, the Net Growing Forward. And basically what we are focusing entirely right now is in control. We are seeing how all these, I'm really interested in our biological control agents here from BC and how they can help controlling the disease. We are working also with the nurseries, trying to minimize the impact of what the nurseries can do to minimize the impact of these pathogens. And particularly, I'm very interested in understanding the host pathogen interaction. What's happening in the plant that some of these fungi are there not causing a problem, which changes are in the plant that these fungi become pathogenic. And we are looking right now to different abiotic stresses, water stress, nutrient stress, overcropping, and see how that may impact so those are also tools we can give to growers. Well, you need to avoid these, when you plant your vineyard, so you are sure that hopefully, you're not going to have the problem. So we do a lot of tech transfer, and that's a big part of our mandate. And I was able to bring the grapevine trunk diseases workshop here to BC. It's the first time it's going to happen in Canada, and only the second time in North America. And this is basically an amazing opportunity for the industry here, not only in BC, but in Canada. I mean, we are going to have the experts from all over the world on this topic coming to Penticton for a week. So I actually opened one day for industry, and what I've done is all management topics are going to be focusing one day. So we are going to invite the industry to come, so they are also exposed to all the research that is being done in other parts of the world. And of course, nothing of this would happen without the funding, without the amazing lab I had. I work close together with Dan O'Gorman. Dan O'Gorman is the biologist, and he's working with me in the lab, our technicians, and all our collaborators, and mostly the great growers. And with that, thank you.
Time for questions.
Yeah.
Hi, I'm a member of the Master Gardeners, and we give advice to home gardeners.
Mm-hmm.
And so I can see somebody asking, somebody from the public asking about this. So to give advice to them, I can see telling them to prune earlier.
Mm-hmm.
That might help.
Mm-hmm.
And I was just wondering whether the
The biocontrol,
that wouldn't
yeah.
be available for the
It's still on the pipeline. I mean, it will take a little bit of time to get
Right.
our own biocontrols
I
available.
see.
But saying that, we are actually trying to to bring, other products available in other places. So I'm actually trying to, we are starting trials this spring, and we are screening different products that are available. One is in the States, so the ones are in Europe. So we are screening here to see if they work under our conditions, and if they work, and hopefully the companies are interested to register the products, so hopefully those products will be available in a much shorter time. It's a different scale, when you talk about, the gardening but we also deal with that. I have a lot of calls in the center, and I get somebody who has 3 or 4 grapes in the backyard, and they want, but, it's a little bit different because mostly those, they are more reluctant, I understand, to use pesticides or fungicides,
Yeah, for master gardeners, we do not.
Yeah, exactly, yeah.
Yeah, we don't.
So hopefully, as soon as we have some of these biological control agents, hopefully they will be available.
But even the pruning.
It can actually help. We know, these are 2 years data we have right now, and it has been consistent that we need to understand right now what really means pruning earlier, under our conditions, because one of the concerns is that we may have our buds more susceptible to frost in the spring, and that's one of the issues that we really want to look at. That's why sometimes, because the buds needs a time to acclimate, for the cold. So it's another thing that I'm not an expert of it, but we have a lot of experts in the research station that they are working with that.
I was wondering if you mentioned that some vines are infected with fungal pathogens, but they're asymptomatic.
Mm-hmm.
Is it an inevitability that the infection will cause damage or necrosis to those tissues in every case?
I don't think so. If not, we wouldn't have viticulture, believe me. Because based on the work we have done, and it's not only my work, but it has been a lot of work done in other places, mostly looking into the status of the planting material, the nursery material, and it has been across the board that we identify and we detect those pathogens in that material. If identification would mean that plant is sentenced to death, we don't see that. So that's why we think there's some trigger factor that is actually causing this sometimes at the major level than others. So we want to understand what it is. Is sometimes issues with water stress. When you plant your vineyard at the beginning, water is very, very critical to get that established. Those roots develop it. That can be a factor for the plant. Is nutrients stress. Sometimes I particularly think that overcropping may play a big role. Usually, it's an advice to let 2 or 3 years the plant growing until we can get a full crop. And mostly under our conditions, where the winters are, hard, and we need to get all these carbohydrates to the roots to spend the winter. The more food we put up, the less it's going to go down. So that's a stress, that it may be weaken those plants. So we are hoping to try to understand this, and that would be another advice that hopefully we can give to growers.
Any other questions? I have a quick question.
Sure.
The survey that you did of the grapevine trunk disease that showed rate of infection with age of the vines, one of the first figures, there was a definite positive increase with age, but there was some dots on that graph where there is a 35-year-old stand with basically zero infection rate. So if you tried to figure out what's going on there.
So one of the things I have to say is the symptom survey we did, it was only based on 40 symptoms we could identify. So we didn't cut all those 60,000 vines. So it's very possible that some of those 30-year-old vineyards that didn't show any symptom, they will have infection inside. But the level of infection probably is not enough to cause these foliage, this declining. So whether those vineyards were lately infected. I'm interested also looking to escape vines. Is something that is in some particular blocks, vineyard varieties, in terms of maybe, I don't know, microorganisms, microbiota, any condition. Also, we are trying to look into what the grower has been doing in those blocks, that maybe help to keep, those vineyards healthier. It's complicated. It's complicated. But you're right. Yeah, you're right that, as I said, if all these pathogens will kill everything, we will have viticulture. So, the idea is try to see, in those old blocks that we didn't see so much disease. Why is that? Because we see that infection is probably inside, most likely.
Did you also correlate production with those vineyards?
Yeah, so we have actually, two trials, and we have actually two growers that the vineyards are getting to 20-25-year-old, and they are actually to a point that they have to completely take out the vineyard because it's not profitable. So the tonnage per acre is really has been slowing down in the last 5-6 years. So we have trials specifically in those vineyards, and we identified because of plant diseases. So we are doing some renewal surgery. So we are trying to cut back and bring back new trunks and see if the infection is not even at the bottom. Because of course, if the canker is all the way to the bottom and past the graft union, it's basically nothing. We are trying to identify also the ages that if growers decide to do this work, is because we know eventually, until we don't have good control strategies and pruning and protection, the vines are going to get infected. So in Australia they do that. In Australia, they have actually a threshold that they know that in some regions, after 15, 20 years, you have to do this work. You have to do all this retraining because the infection is so high. So here would be also interesting to understand, under our specific conditions, when this will happen.
Excellent. Well, if there's no questions, then please join me.
Thank you.
Thank you, Jose.
Thank you all for coming.